In this video, we'll analyze the data presented at the AdCom meeting for Biogen's Alzheimer's blockbuster aducanumab. An AdCom is an expert panel convened by FDA to help them decide whether to approve a drug. And as we discussed in the last video, the AdCom slammed the drug and accused the FDA of conducting a biased review, which tanked Biogen's stock. In this video, we'll take a look at the data to see whether the accusations of bias were founded and whether there actually was evidence supporting that aducanumab worked. So to understand the rest of the discussion, we need to explain the studies being discussed. So the documents here that we'll go through refer to three studies. Study 301, the ENGAGE study, which was a negative phase 3 study. Study 302, the EMERGE study, which was a positive phase 3, identical to ENGAGE in design. And then study 103, a phase 1b study that just measured safety and tolerability. So we'll start by summarizing Biogen's case for approval and then discuss the evidence for or against it. The first point that Biogen made was that the futility analysis was invalid. So Biogen had originally announced it was going to terminate its phase 3 program for aducanumab because the drug didn't work. Basically, there was a futility analysis that showed that the study was going so poorly that there was a negligible chance that it would actually work. And it wasn't even worth it to spend the money to continue the studies. The drug looked so bad. But a few months later, Biogen did an about-face and changed their mind and said they were going to submit it for approval anyway. More on this about-face later. The second argument that Biogen made in favor of its approval was that study 302, the EMERGE study, was positive, i.e. that it was strong evidence from a large, well-controlled study that showed that aducanumab was better at treating Alzheimer's than the placebo. So recall that one of the paths to approval is through having one well-controlled study, which they're claiming study 302 was, and then confirmatory evidence. Usually FDA requires that a drug demonstrates itself to be safe and effective in 2 studies, but sometimes they make an exception and just require 1. So if they can prove, if Biogen can prove that study 302 is positive, then that kind of checks off one of the approval requirements. So back to why the futility analysis was invalid. Biogen's main argument was that there was a protocol amendment that was implemented differently across the 2 trials. And the futility analysis did not account for this, so it was invalid. And the result of this sort of weird protocol amendment was that study 301 did not have as many patients who had, quote-unquote, sufficient exposure to the highest dose of aducanumab. So there was some protocol amendment that made study 301 patients not get enough of the drug, whereas study 302 patients did get enough of the drug. And that explains why study 302 succeeded and 301 didn't. If that doesn't make sense, that's totally understandable. It's sort of not really logical or supported by evidence, so it doesn't make total sense to begin with. But I did a previous video where I talked a little bit more about this protocol amendment. So back to why the futility analysis was invalid. Biogen's main argument was that there was a protocol amendment that was implemented differently across the 2 trials. The futility analysis did not properly account for this, so it was invalid. And if that doesn't make sense, I did a previous video that covered this in more detail. But basically, they said that the studies there was something between the 2 studies that actually made them a little bit different. The analysis didn't account for that, so it was invalid. And then in addition to proving that the futility analysis was invalid, they also need to somehow cast doubt on the failed study 301. Right, if you have one positive, well-controlled study and then one negative study with the exact same design, you can't really argue for approval based on just cherry-picking the positive study and ignoring the negative one. So Biogen needs to somehow make a case that the positive study represents truth and the other one there's something wrong with it and you can't rely on it. So Biogen and FDA laid out several reasons why 301, the failed study, was wrong, quote-unquote, and then 302, the positive study, was true. So in addition to the explanation about more patients getting the higher dose in study 302, Biogen also said that study 301 had more rapid progressors, basically people who had Alzheimer's and their disease got way worse more quickly than normal patients. So they said that the results of 301 can't be relied on for that reason. And there were a bunch of other reasons as well, but those were some of the 2 main ones. So then Biogen also said that, as a consequence of this, they know why study 301 failed. It has nothing to do with their drug. And, it was due to study design, weird patient subgroups, a glitchy protocol amendment, not because the drug didn't work. And therefore, study 302 is positive, 103 is supportive, so we qualify for approval based on this approval pathway. And then plus, our drug reduced amyloid beta across all 3 studies. and amyloid beta is a surrogate endpoint, so we should qualify for accelerated approval too. So accelerated approval basically allows companies to get approved if they treat a serious condition based on a surrogate endpoint. And a surrogate endpoint is basically some sort of biomarker that correlates with clinical outcomes, but isn't, a clinical measurement in itself. So oftentimes in cancer, you can approve a drug with a surrogate endpoint like objective response rate rather than measuring overall survival, which is the important clinical endpoint, because overall survival takes many years to measure. Objective response rate, the surrogate endpoint, can be measured much more quickly, so you can get good drugs to patients more quickly. And then to really wrap up the case for approval, you also have to, basically ignore Study 301, which was the pivotal study they did that failed. Even though it had the exact same design as 302, you just kind of have to conveniently ignore it. And you also have to ignore the fact that amyloid beta is not a surrogate endpoint. Amyloid beta has been demonstrated to have no correlation with clinical benefit across dozens of big Alzheimer's studies, including the studies that Biogen did. But, we have to ignore that as well. So in our last video, we discussed how the AdCom called FDA and Biogen out for, ignoring these two inconvenient facts and more generally conducting a biased review that emphasized the data that supported the approval of aducanumab and ignored data that did not support the approval. They basically ignored data that showed that the drug was ineffective. But I made a full video on that, so you can take a look at that for more context. But in this video, the short version of the AdCom is that they nearly unanimously rejected aducanumab, and they rejected the FDA and Biogen's assessment that the drug was safe and effective. And they also accused the FDA of ignoring its own reviewers. But the FDA did conduct a more unbiased analysis, and its conclusion from that unbiased analysis was that the drug could not be approved without another study. However, the FDA buried this analysis in the appendix, and FDA didn't mention it at all in the briefing document. So this brings us to Appendix 2, the statistical review. So if we go here, actually can't click on that. So 246 here is the statistical review. And again, you can't actually select the text here. So this was done by the FDA's statistical reviewer, Tristan Massey. This was in addition to the clinical analysis and the other appendix, one of the main internal analyses that FDA did. And I highly recommend just reading the executive summary here of the statistical review. It's fairly short and it's sort of written in a little bit of a rush style, but it does lay out good counter arguments and a more unbiased perspective on the data. And some of the quotes are uncompromising, as some of Dr. Emerson's comments are as well. But just to hit a couple of the highlights here. So in the executive summary, basically they say that there's a lot of inconsistencies and issues with this clinical efficacy data. So there were 2 phase 3 studies, but they were both terminated for futility. So the NDA package doesn't contain a single phase 3 study that was fully completed according to the plan. So usually that's sort of a non-starter for FDA. If you don't finish your study, they're not even going to consider it. And in this case, almost 50% of the patients were missing the week 78 time point of the CDR-SB, which is the primary endpoint. This is a rating scale to measure the progression of dementia. And that week 78 time point that had 50% missing data was the only time point that showed any statistical significance, and it was only significant in one of the studies. So what he means here is that this is the change from baseline on the CDR-SB scale over time. So the week 78 time point of this metric is the primary endpoint for both of the studies. And there's no statistically significant change in this prior to week 78. So here you see there's no statistically significant difference between the high dose or the low dose and the placebo. But then between week 50 and week 78, the high dose arm does a lot better and does achieve a statistically significant improvement on this metric versus placebo. But because we stopped for futility, 50% of the data were missing at the time of this analysis. So in study 301, the partner phase 3 study, there was no difference between the high dose and placebo or the low dose and placebo at any time point in the study. So the review then explains that this one positive result, this one statistically significant difference, is still explainable under the null hypothesis that the drug has no effect. So if you do 2 studies, then there's a 9.75% chance that there's at least one type 1 error. He also says that if someone has 2 studies and you take the best study and pretend like it's the only study, then your estimate is most likely biased and misleading. That's a fairly damning statement, and it's not inaccurate. So a type 1 error is a false positive. So based on the statistical design of the studies, there's a 9.75% chance that one of the studies would be positive and showed that the drug outperformed placebo, even if the truth was the drug didn't work. That's just how the statistics of these studies work out. There's randomness in them. It's a sample that's not representative of everyone in the world with Alzheimer's. All sorts of other reasons why you can have these type 1 false positive errors. But as Dr. Emerson said, the issue was that FDA did not take the position that the null hypothesis was true, that the drug had no effect, and that this could just be, the positive study could just be a false positive. They didn't take that position. But there's a reason why 2 positive studies is a standard in Alzheimer's. Because there's type 1 error in all studies, but also specifically for Alzheimer's and these neurological diseases, the endpoints are subjective and have more variability than more objective endpoints like mortality. So the reproducibility of any given study in a neurological indication with subjective endpoints, the reproducibility is going to be lower than most other studies. So you need 2 positive studies as the standard. The review then says that the approach that was laid out in the briefing book, and the approach was criticized by the panel, wasn't justifiable because it was biased, right? It's most likely biased and misleading. And it mentions that the only valid analysis was the only valid analysis of 301 was the pre-specified analysis. The whole point of pre-specifying an analysis is so you can rely on that analysis and you design the study so that you can be confident in the results of that analysis. These post hoc analyses are not justified. And then for the rest of this executive summary, a lot of this dismisses this rationale that FDA and Biogen presented. So there's all of these little data points that FDA mentioned in the post hoc analyses as being a reason for discounting study 301. And then he basically says that, like, look, if you're going to cherry pick positive data, I can go ahead and cherry pick negative data. And then lays out a ton of counterpoints to the FDA and Biogen's arguments as to why study 301 was actually potentially a better study and why 302 is not reliable, and then points out all sorts of other inconsistencies in the studies and the analysis. Things that if the drug was actually effective and the FDA and Biogen's post hoc analyses were actually true, these data points just simply would not exist. So as a side note, the whole debate here between the panel and FDA and Biogen and everyone, this is why you have to pre-specify your analysis plan and then design a good study to clearly test a specific hypothesis. Otherwise, you can just dig through the data and cherry pick observations to support pretty much any given view. And then you end up having these circular discussions that can only be solved by designing and running a good study, which they actually already did here. But FDA and Biogen just chose to ignore one of those studies. And then finally, the statistical reviewer concludes that there is no compelling substantial evidence of treatment effect or disease slowing, and that another study is needed to confirm or deny the positive study and the negative study, which is a conclusion that I think is rational, and many of the panelists believe that as well. So in addition to all the big issues concerning the process that were mentioned in the panel, there were plenty of concerns with just 302. So just taking on face value that 302 is a positive study is not something that the panel was prepared to do. So the next 2 slides will lay out some of the quotes from the panel regarding this. And then also from the statistical review that we just discussed, that also lays out many of these considerations. So in addition to all of the big questions about bias and about the FDA sort of pushing the panelists in a certain way or a certain type of discussion, there's just fundamental critiques with the study 302 overall. So here I'll walk through some of the quotes from the panel regarding this. In addition to the statistical review that we just discussed, it laid out a lot of these considerations. So a lot of these points are, somewhat detailed statistical points. And while it may sound like, unimportant quibbling about technical details, these statistical details are critically important. So these are like the difference between a failed study and a successful one. And these details also are the difference between an honest portrayal of the evidence and a biased review. So the whole point of this AdCom is to bring in experts to evaluate these technically, idiosyncratic but highly, highly important details, because the devil truly is in the details with these sorts of studies. But I'll start with a point that's just a little bit more straightforward. Even if there is a statistical benefit, even if aducanumab has a statistically significant benefit over placebo at that one time point at the primary endpoint, it is a clinically meaningless benefit. So Dr. Alexander, one of the panelists, mentions a study that shows what constitutes a clinically meaningful, important difference for patients. So these schedules, things like CDR-SB, they measure on a scale of like 1 to some other number, and like how do these changes in these CDR-SB and other metrics actually translate to clinical care? So there's a lot of literature on this, and it's generally understood that a change in the CDR-SB of 1 or 2 points for patients with mild to moderate Alzheimer's is the minimum level for clinical improvement. So if it's less than a 1 or 2 point improvement, it's not clinically meaningful. And as we see here, Biogen only saw like a 0.4 to 0.5 point improvement. And again, this was the only time point that showed statistical significance. So you compare this, very small 0.4, 0.5-ish clinical improvement to the 1 to 2 point improvement you need for clinical benefit at a minimum. And this just, suggests that even if this is a statistically valid finding, it's not clinically important. Right, so even if you ignore all the statistical concerns, ignore the missing data, ignore the fact that there's only one time point in these 2 studies that shows significance, ignore all the cherry picking and everything, the magnitude of clinical improvement showed with the single best data point was just clinically not meaningful. Another concern is that there's no correlation between biomarkers like amyloid beta and clinical outcomes. So this is Dr. Alexander again. So he lays out a number of concerns with 302 itself, in addition to critiquing the idea that you can just look at 302 alone and ignore 301. And here this point is that there's no correlation between the plaque reduction and week 78 outcomes. So especially if you're trying to get accelerated approval, there's no way you can have a surrogate endpoint, which you need for accelerated approval. You can't have a surrogate endpoint if it doesn't correlate with clinical outcomes. That's like the definition of a surrogate endpoint. And correlation between biomarkers and clinical outcomes, it's not just like, used for accelerated approval. It's also an important tool for helping researchers understand whether a positive finding is real or is a false positive. So if biomarkers and clinical endpoints are all positive and correlated, that increases the confidence that you have that you're seeing a real effect. And this is especially important in studies like neurological studies where you have subjective endpoints. Another major point was that there was a huge amount of missing data. As mentioned earlier, I think up to 50% of patients were missing at the primary endpoint. So again, the study was declared futile, and after that futility analysis, they stopped the study, they brought in the patients for a final visit, regardless of how far they advanced in the study, and a huge number of patients did not make it to that final week 78 visit, which was the time point where the primary endpoint was measured. So this would usually be considered a non-starter by most researchers and FDA probably as well. So they did do an analysis to see how sensitive the conclusions were and results were to missing data, but those analyses had a potentially fatal flaw, which was they assumed that the missing data were random. If the missing data weren't random, that would entirely invalidate this analysis, and it would invalidate the positive finding on the primary endpoint. But there was no discussion of this critical assumption. Additionally, there wasn't a consistent effect across subgroups. So even in the positive study, you didn't see a consistent effect across subgroups, which is what you'd expect to see with the strong efficacy signal. So if there's a real positive result, you would expect that to be robust and consistent across subgroups. And seeing this consistent effect is another key tool that researchers use to increase their confidence that a positive finding is real and not a false positive. And again, these are extra important in studies like these with subjective endpoints. Which brings us to the next big issue with 302 is that up to a third of patients were unblinded. So this is, again, Dr. Alexander, that up to a third of patients had a mid-dose study increase and more unblinding or potential unblinding. So another panelist elaborates on this concern about unblinding and bias. So 35% of patients in the study were exposed to, they had an adverse event called ARIA. So basically, 35% of patients got diagnosed with this, and if you get this weird side effect called ARIA, it's unique to aducanumab and other similar drugs. You'll know you're getting the drug and not placebo. Like you don't get this side effect if you get a placebo, and you can't miss this. If you're diagnosed with this, you have to go through intense MRI surveillance every month. So Importantly, this is an objective endpoint that's measured, or a more objective endpoint that's measured through MRI. And for objective endpoints, you don't have as much concern about the placebo effect. The placebo effect is worse with subjective endpoints. So you can't really miss this either. If you get diagnosed with this adverse event, you have to go through intense MRI surveillance every month. So there's really no question amongst the patients and caregivers. If you have this side effect, you know it, and you know you got the drug. So this is problematic because the primary endpoint and all the other major endpoints depend on subjective information provided by patients and their caregivers. And these scales that were the primary and secondary endpoints are very, very sensitive to bias. So 35% of patients are unblinded and know they're getting the drug. And you're measuring these subjective endpoints that are very biased and very sensitive to the expectations on the part of the patients and caregivers. So a very large number of the data points that comprise your primary and secondary endpoints are subject to bias. And there's a high likelihood that these are open to bias because of the unblinding. So again, this may sound like an esoteric statistical detail, but this is like critical to the interpretability and reliability of the study. Subjective endpoints like these are a main reason for the reproducibility crisis in neurology. They're super unreliable endpoints, and even more so when it's a known subjective endpoint and there is a lot of unblinding. So especially if you're trying to get accelerated approval, there's no way that you can get accelerated approval if you don't have a surrogate endpoint. But also you can't rely on one study and confirmatory evidence if you have a study with low positive predictive value. So in this, the finding is an underpowered study because there was so much missing data. And when you have an underpowered study, it decreases the positive predictive value of a positive result. So this is a statistical point I think brought up by Dr. Emerson. And a lot of times, if you see a small study with a big statistically significant result, a lot of people think, oh, there's a huge effect size, because otherwise this small study wouldn't have statistical significance. If there's a huge effect size, the drug must be working. But that's not the case. So false positives happen with underpowered studies, in addition to properly powered studies. And if a false positive happens with an underpowered study, you mathematically have to have a large effect size. Otherwise, you wouldn't get a statistically significant result. But having a large effect size does not mean it is real. A large effect size is not a real effect size, especially if the positive result comes from a study with missing data and biased analyses. So while some people may say, and Biogen may say, that they had an underpowered study because of missing data and it was still positive, so the drug must be working. But that's simply not true because the positive predictive value of that study is low. And that's why you need confirmatory studies in the first place. Anywhere between 20 and 70% of phase 3 studies confirm positive phase 2 studies, which means that 30 to 80% of these phase 3 studies don't confirm the phase 2. There's just tons of false positives. And you need something with a high positive predictive value if you're going to rely on like one big study and confirmatory evidence. So that's just another really big concern that I think and the reviewers think undermines the case for approval of this drug. So with all these facts in mind, it's the more likely explanation for the fact that 302 and 301 had discordant results. It's more likely that 302 was just like the one false positive study out of dozens of amyloid beta studies, and that the drug actually doesn't work. That seems entirely plausible. And considering the total body of evidence, that seems a much more likely explanation than the explanation that, 302 was the true study and 301 was just like some sort of fluke. It's hard to see that perspective as the true one in light of the evidence. So another endpoint here is that another point is the secondary endpoints were not interpretable. This is another more technical detail. But essentially, we can't actually interpret the secondary endpoints in these studies. If you recall, seeing correlation between primary and secondary endpoints is another way to increase confidence that a positive result is a true positive. And 302 did appear to have positive secondary and primary endpoints, but because of the way that the statistical analysis plan was designed, if the low dose fails, you actually can't interpret the secondary endpoints. So, you can't actually trust in those findings that the secondary endpoints were positive, and you can't really view those or interpret those as supporting the primary endpoint demonstrating effectiveness either. So there's a mountain of evidence against the drug here, but FDA approved the drug anyway, over the objections of the AdCom. And actually, 3 of the AdCom panelists resigned because of this review. They just said, if we're going to be on these panels and just ignored, like, why would we even take part in this dog and pony show? And this was one of the most controversial drug approvals of all time. And the stock jumped. Biogen stock jumped, as you would expect, on this unexpected approval. But as you can see from the rest of the chart, that isn't the end of the story. The Biogen stock is actually trading around the level that it did after the original futility announcement. So in the next video, we'll wrap up the aducanumab story and explain why the stock, trade down here, and also why that may not be the final chapter for this crazy drug. So thanks again for tuning into this. And if you want to hear the rest of the story, tune in next time. If you have any questions, let me know in the comments.